Joe, thanks for joining us today.
Good morning, thank you. Yeah, glad to be here.
So what are the main analytical methods you use when analyzing biosimilars and comparing them to their reference products?
So many of the methods that we're going to use for biosimilars are going to be the same that you'd use for a traditional reference product. They're the classic methods that we've been using for decades. You're going to want to determine what the structure of the molecule is, so you're going to use some type of protein sequencing. You're going to want to determine what kind of glycosylation patterns are on. Most antibodies are glycosylated, so you're going to want to determine those patterns. You're looking for post-translational modifications of C-terminal, N-terminal variants. You're also looking for impurities in the process, those kinds of things that are just more standard and traditional. You'll do some higher order structure measurements of a variety of types. So most of the methods that you're going to use for biosimilars are going to be very similar to what is used for the innovator or brand products. There are a few new ones that we might employ because we're looking more at a similarity rather than trying to characterize the material, but they're the ones that are tried and true. The one exception I would say is that many of the products have been developed many years ago and didn't have the same level of current analytical methodology. So we're employing some of the newer methods that may not have been employed when the brand was originally developed.
Mm-hmm. Like such as a UHPLC or a
well,
higher resolution
UHPLC is one possibility, but I'm thinking more of things like traditionally people would look at WCX for charge variants, and now you tend to use capillary electrophoresis instead, just because it's faster and it's a little more high resolution. The UHPLC is used more often today, but really that's just a matter of getting a little bit more efficiency or speed in the analysis. Really doesn't give you necessarily more information.
How do those methods change for different stages or steps in the development of a biosimilar?
So a biosimilar is a little different than most traditional drugs because in most traditional drugs, whether they be biologics or small molecules, you're going to start out with a limited set of methods early on. You're going to be doing some toxicology studies perhaps, and then some phase 1, phase 2. A biosimilar is pretty much being developed initially to go right into late-stage clinical trials. So you're basically trying to employ those methods, all the methods early on in the process. Probably the ones that you focus on first are the ones that are needed to determine what process how you're going to make the process, how you're going to make the product. So you may be less interested initially in methods that would show degradation over time, and more interested in making sure that you're making the right product with the right properties and the right sequence and the right sort of glycosylation pattern, for example.
How can columns with sub-2 micron particles assist in the analysis of the N-glycan profile of a monoclonal antibody, whether it be a reference product or a biosimilar?
Well, glycans are very complex, and so the sub-2 micron particles offer the possibility of getting higher efficiencies or faster analysis. But I really think that that's not, the sub-2 micron particles per se, that's not one of their sweet spots for analysis. They certainly are used and can be used, but the major advantage that they offer is typically speed of analysis. And quite frankly, the glycan analysis, most of the analysis is actually the sample prep. The chromatography is probably a very small portion of it. So they're certainly useful, but that's not where their sweet spot is for analysis.
What about tryptic peptide analysis of monoclonal antibodies? Are small particle columns useful in that? And are fully porous or superficially porous particles better for that?
So again, it kind of depends on what your goal is. The tryptic peptides of these materials are very complex. And so initially you're trying to characterize all of the tryptic peptides, or as many as you can. You want to make sure that you've got a good coverage of the entire sequence. So there you're looking for high efficiency, and you can get that with either small particles or porous or superficially porous particles. You may have to sacrifice initially runtime because you may have to have a very long run to pick up all the peptides. As you move forward in the program, what the tryptic peptide typically is used for is a tryptic map, and in that case you're not interested necessarily in getting complete separation of all the peptides. You just want to get a fingerprint, and you may focus in on one or two peaks to compare the biosimilar to the the reference product. And there you can use pretty much any column, and particularly if you're going into a QC lab with that, you might use a somewhat larger particle and use a more traditional HPLC if they're not available in a QC lab.
What about the newer particles that have been developed specifically for protein analysis that are superficially porous but have larger pores? What kinds of results have you seen using those?
So I've seen some pretty interesting preliminary results on that. Generally in the development of either a reference a branded product or in a biosimilar, one of the key things you want to look at initially is what does the intact protein look like, what is its mass, what is the mass profile of the various glycosylated proteins. Do you have the right heavy chain and light chain? Are there variants in the heavy chain and light chain, which there often are? And so these small, superficially porous, wide pore columns could be very useful for that, because traditionally what people have done is they've simply run these through a reverse phase column, and they get very little, if any, separation of the different variants. And they run it into the mass spec, and people can use the mass spec to deconvolute the various different peaks. But the problem with mass spec is that it's not really as quantitative a technique as LC. And in addition, if you're looking for small amounts of the variants, it sometimes can be hard to pick them up. If you're looking for 1 or 2% of a light chain variant, for example, in a 99% of the correct material, it can be more difficult to pick up. So I think where those particles that you mentioned might be useful is in actually doing some chromatographic separation prior to going into the mass spec. It'll make it easier for the mass spec to confirm the correct masses.
So we're talking about mass spec. How important are the higher resolution mass spec instruments to this work?
Oh, I think they're very important. I mean, in our lab we have, I can't tell you how many we have, half a dozen, 7 that we use pretty much around the clock for all of our work. And I think they're very important. I think as we heard in one of the talks yesterday here at PittCon, one of the problems with the mass spec is that sometimes they tend to ignore the separation efficiencies, and while the mass spec can be very useful, the more separation you can employ prior to going into the mass spec, the better off you're going to be able to analyze both sets of data.